Thanks, Helen, and thanks for the invitation. It's my first time here at the Newton Institute, so it's been lovely so far. We were having breakfast with cows walking past the cairn, which doesn't happen every day. Well, it probably does happen every day here, but for me it was quite unusual. So I'm going to talk to you a little bit about some actually old work where we were trying to think about virtual clinical trials, or as we call them, phase I trials, and then also bring it bang up to date with some work that's sort of leveraging that tool set, and also sprinkling some other little bits in between. So that's Darwin, and he'll become relevant in a minute. So as Helen said, I run a department inside a cancer centre. As far as I'm aware, it's the only maths department inside a cancer centre, and we're a diverse group of scientists that kind of come from mixed computational backgrounds. There's physicists, there's engineers and computer scientists, mathematicians, of course, and we also have a couple of evolutionary biologists. But everything that we do is what we call multi-PI. So it's team science, everything that we do, because I don't do experiments, my colleagues don't build models, and the clinicians don't do either of those two. So each project has that sort of integration of those three main components. And that really drives a kind of interesting dialogue. And that's something that for the last 15 years that we've been at Moffitt, we've been trying to hone the best way to have that conversation. Because obviously crossing disciplines is something that we all try to do, but it's not that easy, especially when it comes to clinicians. So for the last 13 years, we've been running an annual workshop that I'm going to give a shout out to, and there'll be the newest one will be advertised shortly. And the workshops really are all about bringing clinicians and experimentalists together with modelers, a new generation of modelers. Often we get young folks applying, PhD students and so on, but we've had department chairs apply too. And essentially the whole goal of that workshop is to come up with a question relevant to the theme, to work as a team to try and tackle that question, and then you give a presentation at the end of this very intense week of work. And that's what it is, it's work. It's not like a workshop in the typical sense where you don't actually do that much, but listen, this is very active. And that workshop leads to then the winning team. There's a team who's picked that wins, and that's from judges. And one of the judges is actually a patient. The winning team gets 50K to continue that work. And so those workshops over the years, 5 teams for each workshop, have produced some kind of really interesting science, novel collaborations. Not all of them worked out, but a good chunk of them did. So if you're interested in that, come and talk to me. So I guess I thought it would be useful to think about why we'd want something like a digital twin, and what is a digital twin? Or from my point of view, a virtual patient or a virtual patient cohort. And there's this dichotomy that's kind of become very apparent to me working with clinicians and experimentalists. In preclinical studies, so in animals and cell lines, our whole goal is to come up with the tightest, most reproducible result, where the error is minimized, the variance is minimized, and we believe the result if the error bars are small. And of course, that's the way science is done, because it's got to be reproducible, it's got to be believable. But as soon as you go to the clinic, all of that goes out the window, because there is no error bars, right? There is the population and its variation. And that intrinsic variation actually is really important to why that drug will work or not work in that population. So one of the things I think that's a real opportunity for mathematical modeling is to try and bridge this preclinical homogeneity with the clinical heterogeneity that's the reality that's there. And of course, that clinical heterogeneity is at the population scale, but it's also at the patient scale too, because tumours are heterogeneous, right? So how we think about that, I think, is something that's really important. And of course, a patient's life history is also very different from another patient's, right? And so the clinicians don't know that life history, they just are seeing what they're measuring today. But how you've maybe lived your life, how your immune system has been responded or have been tweaked over the years is also potentially going to have a big impact on your therapies. And so I'm going to talk a little bit to this. Then the other thing is, where does mathematical modelling fit into that kind of dialogue? And I think, truthfully, it fits in every stage. And so, you know, I'd sort of have that context from my title, from board to bedside and back again, because I really think modelling can help shift the theory of the model from with the experiments to the clinic. But importantly, that isn't a one-way dialogue, because that clinical data could have shaped the model, that could have driven a hypothesis that then could have been tested in the lab, or the hypothesis could have actually been generated as a result of experimental data, or it could be tested through a mathematical model. And so there's these loops that occur. And in many ways, of course, the ideal thing is where you have a theoretical prediction, then you can test it experimentally. But if you're trying to change how a patient responds to a therapy, if you're trying to achieve success with a therapy, because it works in an animal system doesn't mean it's going to work in the patient. And that's fundamentally an issue that we're sort of realizing in the US right now, because of course there's been a little bit of a pushback on animal experimentation. And you know, for years, any clinical trial that tried to move to the clinic from a preclinical state had to have animal testing. And was that right? Was that the right approach? But it became the standard. And you couldn't speak to any clinician about trying to drive a trial, even if you had really great cell line data and you'd calibrated it with patients, they wouldn't listen. Whereas that's starting to change now. So I think, again, that's a great opportunity for us as a modelling community to start thinking about, can we use our tools as a way to bridge between these preclinical systems and the clinic? Because we're actually very good at curing cancer in animals, but it very rarely translates to patients. And it does sometimes, but it rarely does. So that's something that I think we need to tackle. So I'm going to talk a little bit about some underlying aspects of the work that I'm sort of going to present here today. And one of them is thinking differently about how we dose therapies. Most therapies currently in the clinic, and that's across all stages of the disease, are given at what's called the maximum tolerated dose. And the reason for that is that it's historic in many ways, because there was this understanding that if you gave more drug, you'd get more kill. But of course, we now know that that isn't actually the case, because more drug might actually drive more toxicity, it might drive faster emergence of resistance, and also it might actually decay the amount of kill over time. And not all drugs behave the same way. So, you know, and a good example of this, I'm not going to go into the details, is immunotherapy. That was initially tackled as an MTD type approach, but we didn't know, but what is the maximum tolerated dose of an immunotherapy? It almost doesn't make sense to think about it that way, because it's not a drug in the typical sense. Anyway, maximum tolerated dose strategies, the idea here is that we see these what we call U-shaped curves across all diseases. And often you get amazing responses, the burden goes below detection, you don't see it anymore on any sort of measurement that's obvious, and then keep treating because that's the standard of care, even though they've completely, it looks like, eradicated the disease, and then the tumour grows back while the therapy's on, and of course it's now resistant to that therapy. And so, you know, as a simple sort of cartoon analogy to that, and here really saying that it's the pre-existing heterogeneity that's driving it, but of course it could be other reasons. If the tumour's this sort of bunch of coloured cells and the pink ones are resistant, if I could eradicate all the sensitive cells, all I leave behind is the resistant. And so then I'm treating for no point, right? So Bob Gattenby came up with this idea of what he called adaptive therapy. And it's a really simple idea that talks to the fact that we actually want to leave behind sensitive cells. We want some competition there still present with those resistant cells. And so instead of like giving the maximum tolerated dose continuously, what you're doing is now you're given a smaller dose, what you call the minimum effective dose. You shrink the tumour population a bit, but then you stop treatment. Then you let it regrow, and then once it regrows to the original size, you start treatment again. So just by switching the treatment on and off, you're potentially controlling that population for longer. And you know, many people instantly say, oh, that's intermittent, doesn't really work. And it is intermittent, but crucially it's intermittent driven by the patient's dynamics. So you're turning it on and off as their disease shrinks and as it grows. And so one of the big assumptions about why this works, I forgot to say, is that because the sensitive cells actually aren't paying any cost for being resistant, and the resistant cells potentially are paying a cost for being resistant, when you stop the treatment, the sensitive cells grow back quicker. Now, I know I've got colleagues who've told me that's not the case in the cancer they work in, because they see that when resistance emerges, it grows way faster. But one thing that's always worth pointing out in that context, the disease before treatment and the disease after treatment is in a very different environment. You've effectively cleared out the competition, maximum resources, and so now it can grow freely. So, you know, if you were to compare exactly like for like, would it truly be faster? It might be, it could still be faster, and I'm going to show you some evidence that at least there doesn't always have to be a cost. On the left-hand side here is two breast cancer cell lines, the sensitive MCF7 in green and its doxorubicin-resistant counterpart in red. If you co-culture those in no drug, in sort of low serum conditions, the sensitive cells outcompete the resistant counterpart within 10 days. Comparable here on the right-hand side is two sets of melanoma cell lines. There's the WM164 and its resistant counterpart in blue, and this is to a targeted therapy, and you can see the black lines growing faster than the blue line. But if you look at the 1205LU, the sensitive and resistant have no fitness difference. Okay, so the implication is it doesn't always mean you'll have a cost of being resistant. And I always like to show this sort of little simulation that Jill developed years ago, and it's published in 2018. She's sitting back there, and she's got a poster on some cool new work, so go see that on basal cell carcinoma. But this is a really great visual for thinking about how adaptive therapy works. It's an agent-based simulation, it's off-lattice. We grew these tumours from a small number of cells that randomly have a mix of different sensitive and resistance, but they have a hardwired trade-off where the most sensitive cells are the most proliferative and the most resistant cells are the least proliferative. And when they grow, you can see very rapidly that the sensitive cells dominate the tumour, the white cells, right? On the left, I'm going to give the maximum tolerated dose strategy, and on the right, I'm going to give our adaptive therapy. And the adaptive therapy is going to follow a very simple rule of thumb. It's a 25% adaptive therapy, so I'm going to treat until the tumour population shrinks by 25%, stop the treatment, let it return to its baseline, and then keep looping that. And then you can see the distributions of the different cell types that are present. So more red, more resistant, more white, more sensitive. And so on the left-hand side, you can see it looks like we're getting cure, but very rapidly select for those red resistant cells. And on the right here, you can see this sort of pulsing as the tumour grows and shrinks and grows and shrinks. And if you look closely, you can see there's a kind of yellow population that's slowly emerging. Eventually, that's going to fail too. But you can see the difference in the timescales there. It's obvious, right? So very simple way to think about adaptive therapy. And in this case, it was a hardwired trade-off. There's spatial competition. Those two things might not actually exist in real tumours. So, but it's a useful visual. So there's a whole bunch of theoretical work that went on behind the scenes before we actually deployed this in the clinic. And it was deployed in metastatic castrate-resistant prostate cancer. Metastatic prostate cancer goes to the bones. And so you can see here's multiple patients where they've got kind of lighten up on their bones where there shouldn't be lighten up. And then if you look at a more close-up image, you see that kind of dappled scan where there's too much bone or too little bone all across their torso. And it can be quite painful. You know, a lot of men are going to get prostate cancer. You know, fortunately, most of them will die of something else. But the reality is that once you get to castrate-resistant prostate cancer, there isn't many therapies left. And so in this trial, we were using, at the time, it was a relatively new drug, abiraterone, and it was a very expensive drug, so it cost $20,000 a dose. The copay to the patient was $3,000 in the US. So every time they got a treatment, they were having to pay $3,000, which is kind of insane if you think about it. So if we could give them less drug, and we can give them a better quality of life and live longer, that would be a win. And so in this trial, the idea was to use what we called AT50. It was a 50% rule. And in part of that reason it being a 50% rule wasn't theoretical. It was purely because our clinical colleague who ran the trial said patients wouldn't agree to having anything less than 50%. So like if you're saying your tumour shrunk by 25%, we're going to stop treatment now, then patients don't really respond as well to that as you say, well, we're going to cut it in half, right? And then we're going to stop treatment. They're still not sure if that's a good idea. But when they know that resistance is inevitable, which it is in this disease, so that there's no cure here, right? So this is a case of you're going in to try and just do the best you can. So this strategy is much more about controlling the disease. And within prostate cancer, one of the things that's good about it is that you can actually measure from the blood sample the relative burden that the patient has using PSA. And so this is actually one of the patient's PSA tracks over time. And you can see each point there's a PSA measurement. We're giving the drug treatments there. And you can see for that first dose of therapy brings the patient's PSA down by 50%. And then there's a whole period of time where they get no treatment, and then we apply it again, and once it comes back up. So you can see how little drug they get over the course of a year. And you can also see the kind of dynamics of that specific patient. If we compare it with a different patient on the trial, you instantly see that their drug dosing and their dynamics are very different. So, you know, the idea that everybody gets the same strategy starts to make less and less sense when you see these dynamics. And so you can see here it took us a bit longer to bring that patient's burden down, but then also they had a big long period of time without any treatment whatsoever, and then they go back up. So, you know, that was just a couple of patients. The trial's basically finished, although there's still 4 patients who continue to cycle 7 years after the trial began, but the trial's effectively finished. What I'm showing you here is if you look at the top right corner there, that's the actual patient cohort on the trial. Black is when a patient's off treatment, red is when they're on. Each line, horizontal line, is a patient's trajectory. And of course, they accrue to the trial at different times. They'll come off the trial at different times depending on how they respond. But if you compare that with our maximum tolerated dose patients, you can see very easily, visually, they're very different, right? And we can compare it more explicitly with a Kaplan-Meier curve, and you can see the median time to progression is about 14 months for maximum tolerated dose standard of care, whereas we more than double that with our adaptive therapy, and importantly, we double it with using less than half the drug. So we win on both fronts. So this was the first small trial that really showed proof of this concept would actually work in the clinic. And because it was like, you know, hard to convince our clinical colleagues that we should try this in the first place, we have no samples, we have no extra info from these patients, right? It was just to convince them to do it. So there could be a whole bunch of reasons why this worked and why it didn't work, and we don't fully know, right? So I'm deliberately telling you that caveat because that's the truth. So let me talk a little bit about virtual clinical trials, because we're going to apply that in the context of these sort of adaptive therapies, evolutionary therapies. So many years ago, Jacob Scott, who was a talented PhD student who was actually at Oxford indirectly, did his PhD there, came up with this very simple concept, which, you know, to him was obvious, but, and of course the rest of us hadn't really thought about it. But of course, when he said it, we were all thinking about it in some sense, which was, why are we not doing virtual clinical trials before we do real clinical trials? Why are we not simulating the shit out of that to try and find the best strategy possible for the types of patients that we want to accrue? And it was so obvious, and it was just a little opinion piece. That's literally the size of it. And it was like, well, let's try and turn this concept into reality, and that's where Unjun come in. And so back in 2016, so we're almost a decade now, we published this paper in the European Journal of Cancer, and it was a phase I trial in melanoma, and I'm going to walk you through that paper in part. And, you know, the I there was a little play on phase 1, 2, and 3, I being the imaginary number, you know, of course, mathematicians, we love that. But the whole point was, can we optimize a trial design? Can we come up with a strategy that makes the most sense? can we segregate patients the best way by thinking about these, the variability in what we actually see in the population versus what we see preclinically. And we came up with this sort of algorithm type thing. And of course, that was then, and in many ways, it's not so different from what we do now, but it was a little bit idealized where we build the model first, we do preclinical experiments and calibrate it, and it's all perfect, and then we generate virtual cohorts matching to clinical data, and then we stratify and optimize and then deploy it in the clinic, right? So it was a little bit idealized, and the reality is that some of these steps are very difficult to do, and some of the data doesn't even exist in the clinic, so how are you going to actually do that? But I'm going to walk you through what we did there, because I think as a sort of potential motivator going forward of the sort of things we want to do, I think it's a good example of that. And so this is really bringing you bang up to date before I get into that in melanoma with prostate cancer. And this is work that's done by 3 people who are actually in the audience, so Mark, Jill, and Kit. And the idea was, could we actually have predicted the outcome of that adaptive therapy trial without ever having done it, right? And how would we do that? So the crucial thing to be able to do that would be to actually have data on patients that are continuously treated, which shouldn't be that hard to get, right? So first of all, it has to be frequent enough measurements of their burden, and you want a kind of decent enough time span so that you don't have like 1 data point or 2 data points. So we wanted a minimum of 3 data points on these patients. So we looked at, Moffitt has a database of all their patients. There's 23,000 prostate cancer patients. And then we're like, we only want those that have been given continuous abiraterone. That drops it to 600 patients. And you're like, well, shit, I thought we had 23,000. And then you're like, well, is this data actually usable? Then you look at it and there's like another 150 where it's like, the points don't make any sense, like somehow the disease goes away and comes back in a day later or something like that. And then we now want to filter it according to the trial criteria, the original actual clinical trial. So then that cuts down the number of patients. And then we look at those and we say, like, where are the ones where we can actually do some real fitting to? And that ends up bringing us down to 40 patients. So that's the gritty reality of working with clinical data. But it's 40 amazing patients where we actually have really nice U-shaped response curves for all of them. So now what I can do is take my mathematical model and fit to each of those patients and effectively generate a kind of virtual cohort of their dynamics. So I've got, for every one of those, I've got a kind of set of parameters that describes that patient. And now I'm sort of projecting into this 3-dimensional space. Of course, it is 3-dimensional in this case, but in reality, it probably isn't 3-dimensional. It's a much higher dimensional space. But with that sort of calibrated cohort now, I've got something that represents real patients. I'm going to go and treat this cohort now as if it was the adaptive trial, right? So these patients have never been treated adaptively, they're just continuously treated patients. And when I do that, what's kind of amazing is we get pretty damn close to the original trial. So like, this is our phase I trial here. We get our kind of continuous therapy, we do our AT50, and if you see, you can compare that. Obviously, we've got more patients because it's our virtual cohort, right? We can generate as many patients as you want. But you can see that we're getting kind of slightly underestimating how good adaptive therapy was. But this would have been enough to convince our colleagues that we should actually do this, right? This would have been enough to motivate it. So, but what's even better is that with this approach now, you can start doing smarter things. You can actually look at the patient's dynamics, and we've been developing this approach where we look at what's called the first cycle response. So by looking at how they respond on treatment, the first on treatment, and how they grow back on the first off treatment, I can actually calibrate them to a specific arm in this trial. So if we imagine now there's 3 arms in the trial, and one of them is there's a low threshold for adaptive therapy, meaning instead of switching at 50%, I'm going to switch like a lower level, like 25%. And there's a higher threshold where I'm going to actually let it go further. So if we can now sort of, in some sense, personalize which arm they go on to based on that first cycle. And when I do that, then I can do even better. That's the green Kaplan-Meier than our adaptive therapy. So again, the opportunity there is optimizing a therapy and a strategy based on fitting to historic variability that was already present. And so that's something that I want you to think about as I'm showing you this next slide. So this is brand new, it's not published, it's new. So in terms, coming back to the prostate, we're now in the second generation, and in fact we've just started the third generation of this trial. The second generation, we moved to castrate-sensitive patients because our thought was, if we can extend life for castrate-resistant, imagine we move it to castrate-sensitive and we can extend their life. That means that then we can also do it in the castrate-resistant setting. So this is sort of interim results that have been published. We've still not met the median time to progression, but it's going to be significantly better already because we're already at 55 months. And Jill has done some sort of interesting analysis of the patients who are what we call early progressors versus late progressors. And of course, the bulk of them still haven't progressed. These are still all phase 1 small trials. And if you look at the bottom here, you can see this is a PSA trajectory from an early progressor, and then the patient 105 there is a non-progressor. And just by doing some analysis of their first cycle, of trying to think about, could we know ahead of time this patient isn't going to be a good adaptive therapy patient, right? Could that actually be something we could work out? And that first cycle analysis, that kind of on and off, is something that we're realizing could be a very important tool for us. And if you look at that, if you just look at A here for a minute, all we're looking at is their first cycle response. And you can see if we take their PSA over their testosterone level over time, just that ratio separates those patients from the rest of the herd, right? So the red and the blue are above everyone else. And so being able to actually separate now and pinpoint those patients is something that we're leveraging in the new generation of the trial. And if you just look again at the ratio for our two patients, the 105 versus the 109, you can see on the right-hand side here that that is actually a useful metric. And this isn't new, the PSA over testosterone isn't a new therapy, a new sort of marker. It's something that people have discussed. But looking at it in terms of this dynamic sort of measurement, it really gives us some insights. Okay, so I'm going to talk about melanoma. So that was prostate. We originally developed the phase I in terms of melanoma patients. This is a patient with metastatic melanoma. If you've ever seen anyone with melanoma, normally it's black all over their skin. It doesn't look like this. This is a whole body scan that's been pseudo-colored for the metabolic activity. And what you can see is the brain and the bladder should light up, but the rest of their torso shouldn't, and it's completely lit up, and it's because they're covered in melanoma. But this patient has a BRAF mutant version of melanoma, so they can actually get an inhibitor, a targeted drug that will go specifically after cells with that mutation, and within literally 2 months, their skin looks healthy, they feel like they're cured. But unfortunately, if you look just below the lungs there on the right and further down, that actually isn't the case. There's still some disease present, and of course comes back to that issue we mentioned at the beginning, heterogeneity. And so within 6 months, the disease comes back and it's basically in the same location that it started. So targeted therapies can drive drug resistance, especially if they're given at maximum tolerated doses. So our idea was, can we give them adaptively, and what's the best way to do that? So I'm going to talk specifically about patients who don't have the BRAF mutation. So if you don't have that, what can you do? Well, you can actually be given a more broad-based AKT inhibitor. And this is a fairly old study that showed that autophagy seemed to be related to drug resistance. And so this is like from some clinical data showing that patients who have increased autophagy have worse outcomes than patients that don't. Okay, so I'm trying to motivate why we're going to model autophagy. And so I'm going to look at combination therapy, which is a chemo plus an AKT inhibitor. And I'm showing you here various ways of measuring cells under these conditions, right? So there's a thing called viability, a viability stain. There's this thing, acrid and orange. There's a way to look at autophagy through the microscope, and there's this thing called LC3 puncta, which is a way to definitely verify if autophagy is occurring. And what you can see is, without going into too much detail, the combination triggers the most autophagy, right? Just trust me on that. And there's numerous other nuances, but you're probably, some of you maybe never heard of autophagy. In fact, how many people have heard of autophagy? Maybe about 40%. So autophagy actually is a kind of natural mechanism. It's useful for the cells. It's a cellular disposal, waste disposal mechanism. It often provides nutrients to cells when they're stressed. It's a kind of natural thing, and in fact it's something that when you start messing with its signaling pathways, cells might be stressed and leverage autophagy. They can still potentially continue to proliferate even though they're in this autophagic state. And one of the things, so we're going to focus, assume that there's two different states, and it's not just a hypothesis, there's strong evidence for this. But one thing I wanted to show you is that AKT inhibitor, you can see AKT pathway in the top right there, is involved in the autophagy state. So if I now go in and inhibit that, I'm going to mess with some of that signaling, right? So we're going to talk about physiologic autophagy and quiescent autophagy. And I'm just going to like fire into the model without giving you too much background, but I'm going to show you the nice sort of schematic first of the interactions that are going to occur. And so the crucial thing here is that quiescent autophagy is where you're going to not be able to divide and you can die, but you can recover from that state. The physiological autophagy is where you can actually divide in that state, you can die and recover. And then you've got your non-autophagic state, right? So very simple set of ODEs are going to emerge from this. That is the simplest way we can model it. And so basically we have kind of exponential growth, there's death, there's these switching terms, you know, nothing too crazy. The one crazy term is in our autophagic death in the non-physiologic population, because we had to have a delay, because it isn't an instantaneous process, it takes time for those cells to die. And so we tried to get away without doing that, but unfortunately we couldn't. So it's a little bit messy, but anyway, I'm going to show you how we then took that model, we fit it to some simple data. In this case, it's a cell line, this WM3918 cell line, where we're giving continuous therapy with chemo and the inhibitor, and then we're given the combination therapy. And so that's our initial data, no treatment. So you can imagine how we can measure various components of the system by looking at no treatment, chemo alone, this drug alone, the combination, making very simple linear combination assumptions about those interactions. And then we can fit this to all that data. Okay, so just trust me that we can do that. The different curves here represent the different populations, so the colors should be consistent throughout. So then the idea was, okay, can we now do a bunch of experiments where we sequence those drugs? So instead of them just being combined, can we give one first? Can we give the other before it or after it in various ways? And so that was when the biologist said that's where how we're going to do it, and I just couldn't think how that, I can't visualize that. So I drew a little picture because for me that's much easier to understand how the drugs add together, right? And I'm sure you're all agreeing with me on that. It's much clearer. And so you can see it's kind of like the chemo's given in bigger blocks. You can switch on and off the AKT inhibitor much easier. You know, various combinations of those things. And the idea was that the drugs would be, and for most part, the same amount of drug is given. It isn't quite true because obviously sometimes you give no drug of the other drug. Anyway, that's what the cell line experiments look like, which, you know, seems like a lot, but I guess cell line experiments aren't that hard. I don't know if there's any biologists here, probably shoot me for that. But they're fairly straightforward. Anyway, you can sort of stain the cells and you can see what, you know, at the end of the experiment, and you can just by looking at that to see like how many cells are there, it's clear that 7 looks good, right? You know, and 4 looks terrible, and in terms of that, but we can of course quantify it, right? And so that's what we did. So blue is the actual experiment, but what's cool is now I take the model I don't fit it to this new data, I just predict what those combinations are going to be. And what you can see is in red is our prediction, and we do pretty damn good most of the time. So obviously we don't do so great in 9 and 10, but you can see the error bars are pretty big there because of course there's replicates which I'm not showing you and so on. Anyway, the point was that we kind of convinced ourselves, yeah, the model seems to be doing okay here. It's done not a bad job. Okay, so that's the preclinical work. So it turns out there's a clinical trial, a phase 1 trial at Moffitt, actually was combining those drugs. And there's 2 arms. I'm not going to get into the details of the arms because that messes it up a little bit. I'm just going to keep it simple. So this is a classic waterfall plot. And so if you've never seen one of these before, 0 is where there's no growth. Each bar, pardon me, is a patient. And so anything above the line means the tumour grew on treatment. Anything below 0 means the tumour shrunk. And of course, minus 100 means they're cured, right? Just to sort of position. So we thought, can we take our model and reproduce those outcomes with it and create a kind of virtual cohort that would match that distribution? And the answer is yes, you can. And so this is a virtual cohort of 3,300 patients. Don't ask me why we did that, because I don't remember why. But what you can see is that we can gather each of those different types of populations. So they're, in a clinical setting, you have something that's called the complete response, a partial response, stable disease, and progressive disease. So if there's a clinician in the audience, they'll know that, what I'm talking about. That's the standard thing. Anyway, it captures all of that. Now, of course, just because it looks like it, is it actually like it in terms of the dynamics? And so we can do some statistical measures, and of course, probably you've all heard of the Kolmogorov-Smirnov test, which is a way to see if 2 distributions are similar, right? And we can show indeed they are. So the top one's the real one, bottom ones are virtual one, and you can see that the virtual patients and our real patients kind of have a nice sort of match. But that's not completely sufficient. There's this thing called Lin's concordance test, which is where you sample from the 2 distributions and see if they match. And again, we get a highly concordant population. So just because you can map a model to the variation in a population doesn't mean that's really what's going on in that population, right? So just let me add that caveat. We're assuming that the resistance mechanism is the same. We're assuming that autophagy is the main mechanism that's driving resistance in these patients. And of course, in reality, there's probably a multitude of mechanisms, but that's our assumption. So we can take that model and fit it. But now you've got a model that you fit to a whole patient population. And we had our preclinical data, remember, and we fit the model to that data. One of the things I can actually do is I can compare where those cell lines live compared to where the patients live. And unsurprisingly, they don't actually overlap that much. Now, my 3D visual there is a little bit unreal. It's much more sophisticated, the shape. But the point is that it is actually quite distinct. And this really, I think, talks to the issue now. This is a cell line, not an animal model, but it talks to that issue about the disconnect between preclinical systems and patients, right? So the variability is obviously less in the cell lines, but it's also just the dynamics. Patients' disease is much slower than those cell lines. The way it responds is much slower. It's just different. So now that I have this clinical population and my sort of virtual clinical population, I can actually go and analyze it. And what we did was we realized that there was 2 parameters that could effectively split the 3,300 patients into 4 virtual cohorts that have quite distinct outcomes. And so if you look at the kind of non-autophagic growth rate versus the non-autophagy to autophagy rate, the switching rate, if I plot those 2, I can more or less carve up 4 different populations. And you can see C3 is the biggest. It's got 1,734 patients. And then this is the waterfall plots for each of those. And if you look at the switch point, which is where it goes from growing to dying on the treatment, it becomes worse in each one of those cohorts. So you can see that black line that's very close to the the left-hand side, then it almost moves to the middle by C4. So that really shows clearly how different the outcomes are for these patients. And so now I have these 4 cohorts, can actually look and see, well, why did they do worse? And actually, could I do better for one of the cohorts? Could I change the therapy for cohort 4 because we did so bad? Could we do something different? Maybe one of those other strategies could be better for cohort 4. And so, you know, remember, that was continuous therapy. I'm just mimicking their data, but of course we have intermittent therapy, and we have all these things from the data. And so there's a lot here, and I know I'm going fast. So on the left-hand side is an example of a patient getting what we're calling our optimal strategy. So basically for that cohort 4, what's the best strategy we actually could give to that patient? And I'm showing you the different arms of the trial, no treatment, chemotherapy alone. And you can see our sort of green optimum down there, which is actually an intermittent schedule. Keeps the disease more or less in check the best, right? And you can see that, you know, arm 2 does pretty good though. Now, if we look at that across, as a Kaplan-Meier, across all of the variability in the population, not just for one patient, then it's not as good, but you can still see green does best, dark green at the top there. But they're all not doing too bad over the course of 2 years. But one of the important things that we kind of realized was the actual amount of drug that we'd be giving for that optimal patient ends up being way more over the 2-year period. So there's a trade-off to be had there. Potentially the optimal might not be good if there's toxicity issues. Anyway, I know that was kind of like, I flew through that, but hopefully what I showed you there was the potential of how you can use mathematical models to give insights into what variation that's really in the population and how to exploit it. I'm going to take a slight detour now, still melanoma, and I'm going to come back to the preclinical setting, and now we're going to work with animals. And so this is mice that have been given the targeted therapy, this BRAF inhibitor, the Plexicon drug. On the left, no treatment, continuous treatment, and then a bunch of different intermittent schedules, right? Each line on there is the burden of a mouse's tumor measured over time. And what you can see is quite diverse. So just if you look at the no treatment, you have some mice where their tumor barely grows and some mice where it's like, you know, faster than exponential almost. Anyway, one of the things I always found interesting when I look at this is how treatment unifies the dynamics across the animals. So if you look at not continuous treatment, the intermittent treatment. And I think one of the reasons for that is that you're effectively telling the tumor when it can divide. Because when you block it with the inhibitor, you're stopping division, you're also causing death. You remove that and then now everything can grow again. So it's almost like a way to sort of unify the dynamics. That's not what I'm talking about, I just thought it was interesting. One of the other interesting things about this cell line is it appears to be plastic. I know there's a lot on this image, but I want you to just focus on the left-hand side, focus on the black line versus the blue line. And so the black line is the basal population, the sensitive population, and this is a classic dose-response curve. If you've ever seen one of those, you're sort of increasing the drug concentration on log scale. And you can see that at the beginning, with very little drug, you get 100% growth, and of course, we get very little growth with way much more drug. The resistant population is that dark blue line. And so if I take that resistant population and I now treat it, you can see I've got to increase the drug way more and I still only get a small amount of kill. All the lines in between are actually when I remove the drug from that resistant population for 1 week, 2 weeks, 3 weeks, and so on. And what's happening is that the population's resensitizing over time. Okay, so by, I think, roughly week 5, we're as sensitive as we're ever going to get. We're not going to get back to full sensitivity, but we're kind of getting pretty close. So what this shows is that there's some sort of plasticity in this population, or of course, a small number of those sensitive cells survived and they grew back, right? We didn't do single cell sequencing on this, so I can't tell for sure, but we think it's a very plastic population. And so as a result of that, we wanted to build a plastic model. And so very simple Lotte Volterra system here. Sensitive cells on treatment can become resistant. Resistant cells off of treatment can return to sensitivity. So just with that simple sort of switch, I'm going to model the dynamics that I just showed you. Again, this one now has a kind of logistic term. You know, it's as simple as it possibly can be. So we can actually take that model and fit it to all of the data. These are all the replicates for the different types of, you know, intermittent versus continuous versus growth. What that does is it gives us a parameter set for that model for this cell line, okay? And importantly, this is a cell line that's in an animal, right? So remember, these are animal models. So you're getting quite a bit of diversity in these animals. And so what I can do now is use our calibrated model to make decisions for a new set of mice about when to treat and when not to treat. And so that's what we did. So in a way, what we're doing here is we're creating a virtual cohort of fits to each individual mouse's treatment trajectory. And at the very beginning, our fits are terrible because you've got like a few points, right? So you're basically, it's wide open. But the more points you get on that animal, the better you get at predicting what that outcome will be, and the better you are in making decisions. And of course, this is an adaptive therapy approach we're trying to implement, and the decision was when to start and stop treatment. And so this isn't using a rule of thumb, it's using the mathematical model to decide when to start and stop treatment. And the decision essentially was, if on treatment it does worse than off treatment, then we're not going to treat. So, you know, simple things like that. And so what you can see here is the 10 mice, and I've done it kind of like the original trial. They all get 2-week bolus injection at the beginning as a baseline, and then we stop treatment, and then the decision now is every day we can decide to start treatment again or stop it depending on the state of the system. And if you just look at the bottom three there, it looks like it's sort of 2 weeks on, 2 weeks off, right? And that's not exactly very interesting. But it's worth pointing out that of those bottom three, so the little V there tells you how much the tumor burden changed over the course of that experiment. And so it doubled in this experiment, but for mouse 35, it went almost eightfold increase in size. So it isn't just the size that was dictating the dynamics there, it was how it was changing too. And then of course, if you look at something like mouse 8, you know, basically didn't really get treated again. Anyway, hopefully I can convince you that we did better. And so one of the things that we did was, as part of this, we did a couple of intermittent schedules, and we did our adaptive schedule driven by the model. And so the 2 weeks on, 2 weeks off didn't do that actually that much better than continuous therapy. Our model predicted optimal, which was 8 days on, 4 days off, did a bit better, but by far the best was our adaptive therapy. Now, of course, it's still failing, it's just failing slower, right? So there's all sorts of interesting things I could go into there about what we actually found and who was dominating that population. It's all in that paper. But I wanted to sort of bring it back to the clinical setting. So I've talked a couple of trials that we're involved in. There's a whole bunch that are ongoing, most of which we're driving, but there's two I wanted to point out at the end here, because one of them is in the UK, and that's the ACTOF. It's all across the UK in ovarian cancer, and I don't know if Trevor's here yet. Doesn't look like he is, but he's involved in that. And then the bottom one here is the ANZADAMP, which is essentially an Australian trial that's mimicking what we did in our original adaptive therapy trial at Moffitt. But these are all, they're both phase 2, so they're hundreds of patients rather than 20 patients. But I did want to talk one last little bit, I've got a few minutes left, about that one, 365, generating novel therapeutic strategies based on the Evolutionary Tumour Board. And that's something that I think Mark might talk to, who's in the audience. And so one of the things that's frustrating about working in a cancer centre is clinical trials take years, decades, right? So, you know, that trial's 7 years. Imagine we could actually change the course of a patient's treatment today. And that's where the evolutionary tumour board. So tumour boards are typically where difficult patients are discussed with their clinical colleagues from different disciplines that have always been multidisciplinary. They've just not had any mathematicians sitting on there, right? And so our idea was, well, let's bring some mathematicians and let's bring an evolutionary biologist and think about this through a slightly different lens. And this trial is still ongoing. We've had about 20 patients accrue to it from all different types of cancers. The bottom line is, can we take the patient's current trajectory, look at historic data, and then try and forecast what the best treatment strategy would be for them? And so because of this being, most of these patients are metastatic, we're going for a much simpler model. And this is something that we call this a TGI model. And essentially it's a tumor growth model with a decaying sensitivity to the drug, right? Very simple. It's minimal number of parameters, but you've got multiple lesions. And this is one of the sort of patient fits that we get out of that. One of the things you realize as soon as you start working with clinical data is it's dirty. And what I mean by that is no two patients have the same history of therapies. You can see the amount of different drugs this patient has got, right? So how are we going to compare one patient with another or make a treatment decision with one patient or another? And so that was one of the first barriers that we had to tackle. And then the other thing is I'm showing you on this graph all the METs and their burdens as measured. And of course, initially those burdens aren't measured in a kind of quantitative way. So we had to go back and volumetrically get them measured, which cost a lot of money. Anyway, the things like radiation kicks in, and now the lesion is no longer active. So it was growing, and then it was shrinking, and then it's gone. That was the blue lesion. And then there's 6 lung lesions appeared in this patient. What was kind of amazing about this was us just trying to visualize what a patient trajectory looked like. Just producing that graph was already a revelation for the clinician. Being able to show them the patient's history and all the drugs and all the lesions, that was already like a win for them. But that was our baseline, and that took quite a bit of time to generate. And so one of the crucial things that we need to be able to make any predictions, because you can't make predictions on a single patient, right? You need more data. You can't constrain it in any way. And so most of these drugs, the decisions that they're making are based on data, based on drugs that have already passed significant phase 3 trials, so thousands of patients potentially with lots of data. Maybe they've been given in combination in ways that were never tested in the clinic, in a trial. But then you've potentially got historic patients of the same disease at Moffitt. So what we do is we went back into our Moffitt records, and we now try and find patients for the same cancer who are at a similar sort of decision point, and use this to start building up a cohort of patients that we can then actually compare this to. But I want to emphasize that the compare there is tricky, because you can see how different, just look at the colors of the different drugs that they get, right? You need to know what the names are. And of course, they've got different metastases in different locations. So it's not a non-trivial problem, but I think that's a problem we should be focusing on. That's where mathematics can really help, I think. So I wanted to show you this one little brief case. So that was the patient that I showed you a minute ago at the beginning. And so the question was, should they continue on the current treatment that they have, or should they switch to a different treatment? And so the options were carboplatin plus paclitaxel or cisplatin plus 5-FU. And so we're sort of forecasting on top of the current trajectory, which is the kind of green cone of uncertainty that we're kind of generating, right? So if we just continued with the current therapy versus these two options, and what's clear is A doesn't look much better. It's worse, in fact, because it's growing more. B is compatible, right? But crucially what happens is we got an extra data point, an extra imaging data point while we were waiting for this patient to come back. And that allowed us to narrow it down, and now it looks like, yeah, they should switch. It's actually a better benefit to switch. And so anyway, the point here is more that this is like real-time driving decisions, that we feel can make an impact today, as opposed to waiting several years. Anyway, obviously this was done by a whole host, and I've tried to call them out along the way. Hopefully you saw most of them. Unjung, who's obviously not here, she's at a Korean institute and went on to do her own lab, and she's doing some cool science still. But you'll be able to hear from Jill, Kit, Maxie and Mark. They're all here, so you should talk to them. But hopefully I've convinced you that kind of virtual clinical trials are potentially a way for us to predict novel trial outcomes. You know, something that we could actually be doing before we run real trials, and it could help change how you make decisions in those trials, what cohorts you want to accrue, what maybe you want to exclude. I think these sorts of trials are also a way to map preclinical and with and different types of preclinical data to the clinic, and that kind of cycle in many ways is important. And then this idea of looking at initial, like a perturbation, what we call a first cycle, an initial treatment, is a way to calibrate your model to then make more accurate predictions. And of course, you can keep updating that model over time. And for any of this to work in our approach, we really need dynamic measures of burden. Historically, you know, patients get minimal monitoring during the course of the disease. Now, prostate is one of those unusual ones because it's very easy to get. If this is breast cancer, that's a much harder problem. Ovarian cancer, again, they have some nice ways to measure it. So we're working with colleagues where we're looking at ctDNA as a potential way to measure disease burden, but of course it's a bit cloudy what really ctDNA is telling us, and so one has to be careful about how we interpret it. I think there's a way to use these approaches to optimize individual patient trajectories, but the clinic isn't ready for this, right? So the truth is that in the clinical setting, the idea that you would change a treatment along the course of their treatment paradigm is still not quite there yet. We've got a very unique environment at Moffitt where we can do things like, you know, this sort of tumor board, or we can run these trials. But that's really where we want to go. And once I think the world accepts that, then we'll be, you know, absolutely required in every one of these decisions. And I think then finally, you know, potentially an opportunity there for old drugs that you threw out and you didn't think would be any use, could actually be used in a smart way now in the right sequence. So obviously I have to thank the NCI for funding us, especially the Cancer Systems Biology Consortium. And one final point before I go is, if you're interested in any of this, we have a blog and a newsletter, and Maxie's one of the drivers of that. Completely free. If you're interested in contributing, we'd love to hear from you, but we'd love to see this community grow. So thank you.
Thank you, Sandy, for a really fascinating, stimulating talk. Time for some questions from the audience. Thank you, yes.
This may be too simple a question, but the going back to the point about having these two, essentially you've got the treatment resistant and the treatment sensitive groups of cells. The fact that if you keep the treatment sensitive in some reasonable proportion and then they're out competing the treatment resistant, says that fundamentally they're better as cancer cells except for the treatment resistance.
And you're just saying, is that you're questioning that assumption?
Yeah.
So I mean, this goes
something about
again,
the underlying biology that's different.
No. So like, obviously, nobody wants to carry cancer with them for any period of time, right? But if you're in this setting where there's no cure option, right, and what I'm trying to do is now control your disease. So am I going to tolerate carrying some cancer burden with me if it means I can actually control it? So there potentially needs to be a kind of mental shift in how we think about metastatic disease. We can't cure it, right? So trying to cure it is a pointless exercise. So can we control it? And if you can accept that you can control it for a bit longer than trying to cure it with failure rapidly, then maybe this is a better option. Obviously, bigger tumor burdens potentially have other problems, right? Other side effects, other issues. And potentially there could be other mutations that are triggered or other events that could occur over time. But fundamentally, this is a shift from, you know, cure to control. But cure was never an option. So I'm not proposing this for all stages of cancer therapy, right? This is really in the metastatic setting.
So are there specific indications where you would favour it? Because I think with prostate, it's very long. It's slow growing.
Yeah. So prostate is long and slow growing, and so in the castrate resistant setting, we've moved it now to the castrate sensitive. Basically those patients are just waiting to fail. They're not curing on that therapy, and in the castrate resistant setting, the failure rate's within a year, right? And then there's not many options left after that. So the point there is that if you can fail slower with a better quality of life, then that's more worthwhile. But of course, if you have bone metastasis, that could be a painful option, and they'd rather have a more rapid shrinkage and maybe a few months of better quality of life. That might be true, right? So it's a trade-off for sure.
Eric.
Just thinking about when you were pruning your 23,000, prostate kind of histories from the Moffitt cohort.
Yeah.
I would have thought there would be really interesting information in some of the patients who stop treatment. If you have initial response and then they come off treatment for whatever reason, and then what happens? So why didn't you look at those? Because my naive thinking would be that would have really useful information for parameterizing your model as well.
Great point. Yeah, so I think that we initially just thought we'll look for patients who have the U-shaped response because that will be the cleanest, simplest. But we actually underestimate now what the trial would be. So we're going back and looking at that data and including patients such as those that you're talking about, right? And also patients who maybe didn't rebound, they went flat, for example, right? Or as you say, they just stopped treatment altogether, or maybe they were cured, or, so there's various other inclusion criteria. But in a different disease, in ovarian cancer, we're actually looking at that sort of dynamic. We're looking at patients who on continuous therapy stopped for some other reason, whether it's financial or toxicity reasons, and that's given us useful information for our models, right? Because that's break in treatment, and then they go back on treatment again. We can actually see what the disease is doing. So I agree with you.
So I'm going to take one more question, and then we're going to have a bonus poster not on the timetable. Maybe two if you're quick.
You've got to do it.
Yeah.
It's a really dull question. It's not fun.
Great.
Health economics of all of this. Is it likely to end up being cheaper doing your approach or more expensive? Because that'll dictate the acceptability.
Yes. No, that's a great point. I think that initially drug companies don't like it because they think we're giving less drug. But if the patients live longer, we end up giving way more drugs. So ultimately, it's a win for the drug company, if that's your question.
Yeah.
Okay, so that was good and quick. So I will, if you still want to ask your question, you're most welcome.
Well, more of an observation, Sandy.
Okay.
Where are digital twins?
You didn't see them in there.
So if we define digital twins as a replica of a patient's cancer that evolves alongside the patient, is that a good definition for everybody?
Yeah, I guess.
Then all of it is almost a digital twin.
Yeah.
All of these simulations are digital twins. So is the patient-specific calibration a data problem, or is it a modeling problem, or is it both?
Yeah, I mean, I think that, again, this is a bigger question. I think that we should definitely have a more group discussion. But I think in terms of, the modelling approach you use is going to shape what that population looks like, right? So you first have to make those assumptions. This is my model, and that's what I'm going to use. But, whether you're looking at a population or whether you're looking at an individual, I think that's one of the differences here. So you can map to an individual's dynamics. We all do that when we build models, right? Sometimes we'll do parameter sweeps of those models. Is that then, population that we're generating? I think the crucial difference there is that by looking at the distribution of response from the population on that drug, that's where you're now getting the kind of digital twin or virtual cohort match, right? But it is under the constraints of your model and your model assumptions. So I could have fit all of that data with a different model, right? So it's an important point to bring up. So I think it's both of those problems. It's not, you can't separate the two.
So indeed, I think that's part of what we would like the conversation to be about. It's not 100% clear to me what a digital twin in cancer looks like. so many different diseases, so many different types of data. Getting data is really difficult. And so what does it look like and what can it do? And I think you've kind of demonstrated what you can do with underpinning modelling and how you can personalise it. But I don't think, again, certainly from my own experience, that the digital twin concept is deeply embedded in a lot of the cancer work that we do. And I guess that's part of the reason for the workshop, to open that conversation and see what it should look like and what we can do. So I think let's just thank Sandy and we'll move on to our poster session.